Hello everyone. So this video is the third of a four-part series about gene and cell therapies as applied to muscle diseases. So to go over a little bit of review from the previous video, we have a muscle fiber which is basically many cells fused together. The cell nuclei are indicated in blue, and then it's surrounded by what are called satellite cells, which are shown in green. And we imagine that, the cells, both in the muscle fiber as well as the satellite cells, don't have a good copy of a certain gene that they need to stay healthy. So each cell has two copies of the gene, but if they both have a problem and are causing a disease with recessive inheritance, then both of so that's what the two X's are shown on the cell nuclei and also in the satellite cells. Okay, so gene transfer therapy, and this is what normally is meant by gene therapy that is currently in clinical trials. It adds another copy of a gene the gene, which a good copy which works well and allows the cell to make some needed protein. It doesn't do anything about repairing the mistakes in the existing copies. That's actually called gene editing, which we're going to go over in the next video. Okay, so the transferred gene has a few differences from the genes that the cell already has, besides not having an error. Okay, the transferred gene isn't part of a chromosome, which genes normally are. And there's a couple reasons for this. One is safety, that if you stuck the new gene into the middle of another gene in the chromosome, it might make it stop working, and sometimes that can actually cause cancer. So that's as a safety measure. You just have the transfer gene is just sitting in a little bit of DNA that's in the cell nucleus, but not part of a chromosome. It also doesn't include what the promoter that the gene normally uses. Okay, a promoter is basically in the instructions that tell which type of cell is supposed to make the protein that this gene codes for. And basically every protein, every gene in our body has a different promoter, but for the most part, we don't have a complete understanding. So for gene therapy, we normally only use a few promoters. There's a few promoters known which either cause something to be expressed only in skeletal muscle or in skeletal muscle and heart muscle, and those are what's generally used for gene therapy for muscles. Also, the DNA from the gene is already spliced together, which DNA in our genome, in the normal copies of the gene, is in several pieces, which gets spliced together before the protein is made. What that allows is that you can do different splices to make different versions of the protein to be made. But when you transfer a gene, normally it's already spliced, which means you can only make one version of a protein. So there's a few limitations. Okay, so to give an illustration of that, we have up in the upper right-hand corner an AAV, which carries a good copy of the gene, which we'll denote by a gold star, and then the nuclei of the muscle fiber, and as well as the satellite cells have only bad copies. Okay, now where does the good copy of the gene get transferred to by the AAV? Well, we know that, in many cases, and this is what we're trying to do, it gets copied into the nuclei of the muscle fiber. We probably don't get all of the nuclei of a given muscle fiber to get the transferred gene. We don't get every fiber in a muscle to get the transferred gene. We're not at 100% efficiency. But when things are working well, we can get most of the muscle fibers to have the new gene, which does work correctly, and most of the nuclei in the fiber, because we know, that the muscle fiber can be making close to the normal amount of protein that a healthy fiber would make. What we don't know right now is, which we have question marks on the satellite cells, we don't know whether the satellite cells also receive the gene that's being transferred in. Probably they do some of the time, but we don't know, if most of the satellite cells also have, receiving the gene, or how often that happens. Okay, another thing is that muscle nuclei in muscle fibers don't divide. Once a cell nucleus has joined a muscle fiber, it never divides again. So we don't have to worry about the gene that's transferred into the nuclei of the muscle fiber, whether both daughter cells, as they're called, will carry the gene, because they never divide. But the satellite cells do divide, and so then there's the question, well, is the gene that we've transferred also going to get copied and inherited by both the daughter cells? But it's not, we don't actually know that, because it's not part of a chromosome. Normally what happens when a cell divides is it makes extra copies of every chromosome, and then the two sets of chromosomes get pulled to each of the newly formed cells. So we don't know if there's just an extra bit of DNA. We don't know what happens, or whether the two cells will both inherit. Okay, so the way that the cell division happens for the satellite cells, which are shown in green, is if a muscle needs, the fiber either needs to be replaced or to grow larger, the satellite cells divide in a special way. So one of the daughter cells remains a satellite cell, and then it can divide again, many times. The other daughter cell becomes a myoblast. It can divide some more to become, more than one myoblast. But then the myoblast eventually join the muscle fiber, or if a muscle fiber dies, form a new one. Okay, so if only one of the daughter cells from a parent cell that does have the transferred gene inherit it, then after a few divisions, most of the cells which are descended aren't going to have the transferred gene. Okay, so what's the consequence of this? Well, okay, you can think of our muscle fiber that we showed a few slides ago, and say we gave most of the nuclei in the muscle fiber the gold star, meaning they have a working copy of the gene. Okay, but now say the muscle fiber decides it needs to grow bigger, and say also that the satellite cells didn't get the copy, the transferred gene, or or their descendants don't all have the copy. So you now make a bunch of myoblasts which don't have the copy of the transferred gene. And then when they join the fiber, the fiber now is bigger, it has more nuclei, but none or relatively few of the new nuclei that it's added actually have the transferred gene. So that's one open question as to how long gene therapy for muscle diseases might work. We don't know because we've only recently started to treat people. We know that it lasts the lifespan of a mouse, but that's only about 2 years. So it may be that if a person is treated before they're fully grown, then the amount of the transfer gene and the amount of protein made by the transfer gene will be diluted, and so that won't be as effective at presenting the disease. Also, if it has less protein, there may be a higher turnover of the muscle cells because they don't have all the proteins that they needed to stay healthy. So ideally, what we would want from transfer therapy is if we could magically say that all of the satellite cells have a good copy of the gene, and then when they divide, all of their daughter cells do also. Then each satellite cell that you put a good copy of the gene in could then produce many myoblasts which could join the muscle fiber. We don't know to what extent that happens. So that's kind of an open question in gene therapy for muscle diseases right now. It works, but it does have some limitations. It's not quite the way that it works in nature.